our video presentation. And again, good day to each and every one of you. We are, by the way, the group 9, and we will be tackling chapter 10 of sex linked chromosomes and sex linkage. And without further ado, we will introduce ourselves briefly, starting off with me. I am Angel. I am Pearl.
I am Rico.
I am Alona.
10.1 Introduction.
Previous topic, Mendel's working with plants showed patterns of inheritance derived from gene loci on autosomal chromosome. So one complication on this model of inheritance in animals is that loci present on sex chromosomes is called sex-linked loci, but does not follow the pattern that is present. Chapters covers patterns of inheritance, virus, six links of inheritance.
Figure 10.1.2 shows that the most of the chromosomes in humans are present in two copies. Each copy has the same length, centromere location, and bonding pattern. As mentioned before, these are called autosomes. However, note that the two of the chromosomes, the X and Y, do not look alike. These are sex chromosomes. In mammals, males have one of each while females have two X chromosomes.
10.2 Autosome and sex chromosomes.
Hi everyone, my name is Alona Clarabal. So I'm the reporter of autosomes and sex chromosomes. In diploids, most chromosomes exist in pairs, same length, centrum location, and bonding pattern with one set coming from each parent. So, so the chromosomes interact centrum of males and females. These chromosomes are called autosomes. So, however, many species have an additional pair or chromosome that do not look alike. The chromosomes are called autosomes. So, many species have an additional pair of chromosomes that do not look alike, or these are six chromosomes. So, there are six chromosomes because they are differ between the sex. In humans, males have one of each, with females have two X chromosomes. So in the females have one chromosomes, and then are six chromosomes because they differ between the sex. So in humans, males have one of each while females have two X chromosomes. So the male have one chromosomes. Then females have two X chromosomes. So autosomes are those chromosomes present in the same number in males and females. So ang chromosome na same number of females and males. While six chromosomes are those that are not with six chromosomes were first discovered, their function was unknown, and the name X was used to indicate this mystery. So, the combination of six chromosomes within a species is assorted with their female individuals. So, the six chromosomes, there are species assorted their female and male individuals. So, in mammals, fruit, flies, and some pests, plants, those with two are butterflies. Two X chromosomes are females, while those with E and XY males. So, in mammals and plants and fruit, the there are two X chromosomes are females. And males, the chromosome is XY and XY for males. In birds, mans, and butterflies, males are ZZ and females are ZW. So in birdsman butterflies, for males, are ZZ and females are ZW. So because six chromosomes have arisen multiple times during evolution, the molecular mechanism through which are determines six differs among those organisms. So how do the six chromosomes we have during mitosis? So mitosis we have like mitosis chromosomes is not sister chromosomatic separate in monophase and heterologous sex chromosomes do not separate from one another. Still, in those individuals with two of the same chromosomes or two or with two same chromosomes or homozygotic sex or XX females and ZZ males, the chromosome pair on cigarette during meiosis. So the females heterologate XX. And while ZZ female chromosome pairs on cigarette during meiosis. So as autisms, so during meiosis in XY males of ZW females are heterogametic sex in the sex chromosomes pair with each other. So, in mammals or XX or XY, the consequence of this is that all egg cells the Y carry out X chromosomes. So, the mammals, there are XX and XY consequence or all this that all egg cells. So, egg cells will carry either an X or EY chromosomes. So half of the offspring will receive two X chromosomes and become female, while half will receive an X and EY and become male. So the female chromosome, two X chromosomes become female, while half will have. So for female, XY will receive an X and EY and become male. So, in species with ZZ males, offspring carry AZ chromosomes, while in females, ZW half will have a Z and half a W. It is a popular misconception that the X and Y chromosome were named based upon their shapes. Physically, each looks like any other chromosomes. EY chromosomes doesn't look like AY and any more than chromosomes. A4 looks like A4, the combination of six chromosomes. With a species inserted with either male or female individuals in mammals, fruit flies, and some flowering plants, XX individuals are females while XX individuals are males.
Pseudoautosomal regions on the X and Y chromosomes. Before the X and Y chromosomes diverge into their distinct forms, they were once equivalent homologs behaving like autosomes. Over time, the Y chromosome lost most of its genes, resulting to its reduced size. However, the X chromosome retained most or all of its genes, and despite their morphological differences, the X and Y chromosomes can still be able to pair up together during the meiosis process because they still share certain regions. These regions are known as pseudoautosomal regions or PARS, contains similar genes and allow for crossover events. The term pseudoautosomal reflects the fact that these genes in these regions behave like autosomal genes during inheritance. Alleles of these genes undergo crossover similar to those on autosomes, even though they are located in the sex chromosomes. Clinical implications. Genes within the pseudoautosomal region exist in two copies in the XY males and XX females. Consequently, they are expressed from both the active and inactive XY chromosome. In cases of sex chromosome aneuploidy, such as XXY, XXX, XYY, these genes may be under or overexpressed compared to normal females or males. One notable gene in this region is SHOX, or short stature honeyblogs. It produces a protein that promotes bone growth and which it is involving the growth of a human being. Individuals with normal 46XX and 46XY karyotypes have two functioning copies of SHOX or short stature honeyblogs and exhibit an average height of a human being. And in terms of aneuploidy, those with 47XYY and 47XXX genomes possess three copies and tend to be taller than the average human being. Conversely, individuals with 45X or someone who has Turner syndrome have only one copy of SHOX or short stature homeobox and which contributes to their shorter stature.
Six linkage, an exception to Mendel's first law. We have introduced six chromosomes and autosomes, or non-sex linked chromosomes. For loci autosomes, the allele follows the classic Mendelian pattern of inheritance. However, for loci on the sex chromosome, this doesn't follow because most, well not all, of the loci on the typical X chromosome are absent from Y chromosome, even though they act as a homologous pair during meiosis. Instead, they will follow a six-link pattern of inheritance. An X-linked allele in the father will always be passed on his daughters only, but an X-linked allele in the mother will be passed on to both daughters and son equally. X-linked genes, the white gene in Drosophila melanogaster. A well-studied X-linked gene in the white gene on the X chromosome of Drosophila melanogaster. Normally flies have red eyes. Flies with a mutant allele of the gene called white negative or W negative have white eyes because of the red pigments are absent. Because this mutation is recessive to the wild type W positive allele. Females that are heterozygous have normal red eyes. Female flies that are homozygous for the mutant allele have white eyes because there are no white gene on the white chromosome. Male flies can only be homozygous for the wild type allele or the mutant allele. A researcher may not know beforehand whether a novel mutation is six-linked. The definitive method to the rest of six linkage is reciprocal crosses. This means to cross a male and female that have different phenotypes, and then conduct a second set of crosses, in which the phenotype are reversed relative to the sex of the parents in the first cross. For example, if you were to set up the reciprocal crosses with the flies from pure breeding W negative and W positive strains, the result would be shown in figure 10.442. Whenever the reciprocal crosses give different results in the F1 and F2, and whenever the male and female offspring have different phenotypes, the usual explanation is sex linkage. Remember, if the locus is autosomal, the F1 and F2 progeny would be different either from this process. A similar pattern of sex-linked inheritance is seen from X chromosome. Loci in other species with XX to XY sex chromosome system, including mammals and humans.
10.5 Y-linked genes. In humans, the Y chromosomes has been studied and is known to contain approximately 200 genes which provide instructions for making proteins. Because only males have the Y chromosomes, the genes on this chromosome tend to be involved in male sex determination and development. Sex is determined by the SRY gene, which is the sex determining region of the Y chromosome. This is the structure of Y chromosomes in humans. The Y chromosomes have 59 million base pairs, approximately 150 genes, and identified as sex determining in 1905. The upper and lower part is called the pseudo-autosomal region, or the PAR. These regions allows for pairing and recombination between X and Y chromosomes during meiosis, or the process of cell division that produces sperms and egg. Recombination in the PAR ensures the proper segregation of sex chromosomes during meiosis, which is critical for the production of functional gametes, or the sperms and the egg. The green part is called the sex-determining region Y gene. It plays a crucial role in determining the sex of an individual during development. SRY gene codes for a protein called the SRY transcription factor. This protein is responsible for initiating the development of male characteristics by triggering the differentiation of the bipotential gonads into testes. The presence of the SRY gene on the Y chromosome is the primary determinant of male mammal. Other genes on the Y chromosomes are important for enabling men to father biological children or for male fertility. Many genes are unique to the Y chromosome, but genes in areas known as pseudo-autosomal region are present on both sex chromosomes or in male and female sex chromosomes. As a result, men and women each have two functional copies of these genes. Many genes in the pseudo-autosomal regions are essential for normal development. Although the Y chromosomes is sex-determining in humans and some other species, not all genes play a role in sex determination or Y links, so it could be also came from the X chromosome. Y-linked traits, whilst few in number, do exist. For instance, the Y-linked trait of web toes causes a web-like connection between second and third toes. Web toes, which also called as syndactyly, is a case when the skin fails to separate during fetal development. The defects in the Y chromosome or the mutation could be in the limb development, such as the homeobox or HOX genes, especially HOXD13 and HOXA13. HOXD13 and HOXA13 genes belong to a class of proteins known as homeodomain that contains transcription factors. These proteins are commonly referred as HOXD13 proteins and HOXA13 proteins. Mutations in these special genes containing this protein can disrupt the normal development of fingers and toes, leading to syndactyly. Porcupine man. It is a colloquial term used to describe individuals who have a rare condition called hypertrichosis. It occurs when the skin thickens and gradually becomes darker, scaly, rough, and with bristle-like of goats. It is influenced by various genes across the genome, including Y chromosome, but not solely with it. Since Y-linked inheritance involves the Y chromosome, Y-linked inheritance is passed on from the father to son, since it is only male has Y chromosome. Of course, Y-linked traits never occur in females and occur in all male descendants of an affected male. The concepts of dominant and recessive do not apply to Y-linked traits, as only one allele on the Y chromosome is ever present in any one or male individual. This is, of course, ignoring XYY syndrome, which is a rare chromosomal disorder that affects males. Now, let's discuss the mechanism of sex determination by the Y chromosome. Aneuploidy, extra copies of Y. XYY syndrome, or known as the Jacob syndrome, is caused by the presence of an extra Y chromosome. Males normally have one X and one Y chromosome. However, individuals with this syndrome have one X and two Y chromosomes. Most individuals with Jacob syndrome do not have any noticeable physical differences or health problems. However, some may experience taller stature, acne, speech and language delays, and mild learning disabilities. Aneuploidy, extra copies of X. Extra copies of X will result to the syndrome of Klinefelter syndrome. The male affected will experience low testosterone levels, underdeveloped muscles, and sparse facial hair. Although severity of the syndrome increases, the sex is always male. Additional important observation involve aneuploidy, missing Y chromosome. For example, X chromosome has no Y, results to Turner syndrome, having 45 chromosomes. Females will be affected, having short, slow growth, and heart problem. Conclusion about human X and Y. From those discussions, it concludes that it is not the number of X, but the presence or absence of Y that determines sex. Therefore, Y acts as a dominant determinant of male sex.